This is Dr. Charles Parker, and you're listening to Core Brain Journal. It's the place where I connect both fresh discoveries and interesting different perspectives from advanced mind science with the realities of real people and everyday life down on Main Street. Well, welcome, gang. This is Dr. Charles Parker, and you are going to have a real pleasurable time listening to this gentleman out here. Dr. Bill Miller, an MD, is going to tell us about some words that you have never heard before. I haven't heard them myself, so many of you haven't heard them. And he's going to take those words and make them practically applicable in our everyday lives. Bill, thank you so much for joining us. I really appreciate it.
Well, it's a real privilege. Thanks for having me on the show.
So I'm going to read his bio a little bit, and then this will whet your appetite. But some of the words that are absolutely relevant are hologenomic, hologenomic entanglement, get that. And then this is the one I love, Metadarwinism. I'm thinking it's so cool. You know, we're interested in metacognition over here, Bill. I thought I was going into a deep, meaningful new area when I was talking about metacognition, but you're into Metadarwinism. So this will be fun. So let me introduce you. So Dr. Miller is the author of The Microcosm Within, Evolution and Extinction in the Hologenome. He's a practicing physician, if you can get that. He sounds like he's a PhD in the history of mankind. Practicing physician for 30 years. Now he's an independent researcher in evolutionary biology. He's a commentator on health issues, the microbiome, and the evolution of mankind. He does this on radio and TV. And he is the co-editor of the Society for the Advancement of Metadarwinism. There you go, folks. Now fasten your seatbelts if you're in the car, because we're going to have a conversation about all this. And Bill, thank you so much for joining us. Now tell us a little bit about your personal life so we can get a picture of who you are.
Well, first of all, I want to say I loved what you just said about me. Those are all true things, but you didn't mention that I'm also a lousy golfer. Despite all of those nice things, I still can't do some things well at all.
I didn't know that one.
Here's what happened to me. I was in practice for 30 years, frontline physician, loved the field. And I began to notice certain things about the way disease patterns rolled out. And they just struck me that in my particular practice, we were making predictions about who would have what kind of a microbial infection. Meningitis is different from pneumonia. And we would predict the microbe. And I began to wonder why, when you look at imaging studies, the patterns of the diseases were familiar enough, repetitive enough, that you could make predictions. And so what began to percolate within me is the why. Surprisingly, in medicine, we never ask the why. All we care is that we know the association.
Yes.
That's medical practice, it was for me.
Quite so.
I wanted to delve into the why, and that got me thinking about cognition, thinking, who thinks, what thinks. And the thing that shocked me as I looked into it, microbes are cognitive. They have intelligence. This is a tough thing even for physicians to think about, Charles, that microbes are cognitive. Every single living thing has some basal level of cognition. They're not thinking like you and I do, extremely differently. But it matters to evolution, it matters to our health. And we're going to talk about some of those things.
That is so darn interesting. It really is. I'm looking forward to talking to you. So let's talk about how you made that switch in your life. You were talking to the guys at the shop, and you said, I'll bet this person's suffering with staphylococcus. Or how'd that happen? What happened?
Well, the thing was that I knew that a lot of my practice was based on the fact that certain patterns were repetitive. Then I began to understand that they're repetitive because microbes, the things that infect us, which is a great deal of all of medicine, even those things that we don't understand yet, all of those things are repetitive because of microbial intelligence, because cells are intelligent. And then I started to think about Charles, they're just strange things. Here's a revealing anecdote. I went with a friend to a medical meeting in Chicago. The biggest meeting for our subspecialty was in Chicago, and I couldn't stand it anymore. I played hooky one afternoon late. After a long day of meetings, I went to the Field Museum, and it all started with a gal named Sue. It was Sue, the Tyrannosaurus Rex, right in the rotunda, the central rotunda of the Field Museum. And here is the thing that struck me. I'm looking at this magnificent dinosaur, and it is got vertebrae that look a lot like ours. It has a femur, a humerus. These are the bones of our arms and legs, a hip socket, a pelvis. They all look very much like human anatomy. Even the muscle insertions looked correct. And then it had these little tiny silly arms. I don't know if anyone has seen a Tyrannosaurus Rex specimen up close, but when you do, those arms make no sense at all. Then the thing was, I looked at the description, and it said, this dinosaur roamed for between 6 and 8 million years, unchanged. That made no sense. I was blown away. I couldn't understand it. I turned to a partner who was with me. I expressed this to him. He said, you're an idiot. It just takes a lot of time. You're a fool. And I never let it go from that moment. That led me to a journey, a path of discovery that I found was very different. This Metadarwinism term that you utilized. This is a short-term means of discussing theories of evolution, constructs of evolution that are different from standard Darwinism. And here's the important thing for all of us with respect to ourselves and our healthcare. Once you start to think in that way, you learn that although Darwinism honors competition and randomness, what really matters in evolution is collaboration and directed purposes of intelligent cells and microbes working together. Why do we go there? That's the term hologenome. That's what it all means. Every single person that's listening, every animal that they can see with their eyes, are these hologenomes. That means that they are a combination of microbe and internal cells. Let me give you an example. When I look in the mirror, I see myself, and of course I think I'm unusually handsome. I mean, Brad Pitt,
and you are, my man. I'm not flirting with you, but you look good. That's good.
Brad Pitt should eat his heart out. But the fact is, I think of myself as a single being. What else can I do? I'm looking in the mirror. I'm seeing me. Anyone else who does that does the same. That's wrong. It's an illusion. How are we really constructed in the natural world? We are a constellation of trillions of cells. And here's the shocking part. The majority of them aren't our own cells. The majority of the Bill Miller cells are outweighed by the enormous greater fraction of microbes, where bacteria, viruses, fungi, they inhabit me. They in partly control me. This is what we're learning about the microbiome and this concept of the hologenome. The hologenome means you got to think about all the genes of the microbes and the regular cells. The microbiome is the term that we use for all of these microbes that are in us and on us. What we didn't understand is that we can't live without them. They keep us alive, and they're essential for our metabolism and even for the function of our brains and our moods.
That is unbelievable. You know, so many things come to mind right off the bat because I've evolved slowly but surely. I'm not where you are by any means. I'm so happy to hear this. But what we've been seeing in our practice is how these cells do talk to each other and defend against our work in an attempt to eradicate them.
That's right. What we've learned about ourselves, and this is really new stuff. Your listeners and viewers are really getting new, truly cutting-edge science here, and it's all peer-reviewed. This is not just me speculating, but it's still so new that it is largely unfamiliar to the medical community. Here's how we work biologically. We work on a cellular basis in tissue ecology. So people are used to thinking about the natural ecologies. We talk about rainforests. I live in Phoenix. That's desert ecology. The Northeast is different with pine trees that we don't have here, and so on. So people are accustomed to thinking about the outer world as ecologies. My point, and this is absolutely true, is that all of us are a collection of ecologies ourselves. Each body tissue type of ours, our gut, our respiratory system, our brains, our lymphatic system, they're all forms of ecologies, and they link together. They work together, they compete together, they collaborate together, and they combine so seamlessly that when I look in the mirror and I see me, I think I'm one thing. But nature deconstructs us into our individualized segments with respect to immunology, response to stress, response to infectious diseases. That's getting back to why do some infections go to some tissues? Why does pneumococcus go to the lung? Why does meningococcus go to the nervous system? Because that's their preferred environment. Those are preferred habitats. So they do this reliably enough that we can make predictions in medicine and protect people and provide the proper antibiotic even if we can't culture it.
That is so interesting, because I really haven't even thought about that before. But you know, one of the things I do in my practice is, I'm a psychiatrist. We work with brain, and we work with the whole connection of brain and body, which is a very primitive way of thinking about what you're talking about. But we see people who have, for example, candida infections, bowel candida infections. Mm-hmm. And those bowel candida group together and form biofilms. So the question, and it sounds like what you're saying is, candida itself has a specific, could have a specific interest in bowel colonies that would drive them to go there.
Exactly. This is the part that's very hard for people to, not people, I'm talking about doctor people, and scientists to accept. But now it's getting to be quite well understood. In cellular life, there's a thing called homeostasis. It's a fancy term, but what it means is a net state of preference. Mm-hmm. The thing that the living organism, the cell, what is its preferred status? Its net metabolic status at the moment. What is it like? And we don't think about microbes as liking environments, but that's precisely what they do. Candida is going to the environment of that it can, because it wants to. That's the part that is almost frightening. The purpose of the microbial world. It is flexible. It is purposeful. And it has consequences for us when we start to think about our microbial companions in this entirely new way. This leads to a whole new revolution in medicine. This will lead to effective treatments, and some of these are going to be microbial transplants. That's actually already being done in real time. This is no longer science fiction. This is the next wave of medicine. You know, medicine has had a number of truly dramatic waves. First, there's those genuine anatomy through by dissection. Then let's say there was anesthesia. What a revolution that was. How about imaging? How about Röntgen and X-rays, and then all the forms of imaging, MRI and ultrasound that we have today? There was antibiotics, antisepsis. Each of them was a huge revolution in medicine, which has translated into enormous improvement in health. Vaccination, another great example. Here's the next revolution. This one is now without question. Learning how to manipulate our microbial portion, our microbial fraction, to our betterment. And those experiments are already undergoing, and they're already showing real-time pretty good results. I mean, of course, nothing's perfect, but it's being used all the time. Things like fecal transplants are being used now to treat inflammatory bowel disease. We're going to learn to manipulate ourselves in a new way, and it's going to have a dramatic effect on people's health. Take diabetes. We're relearning all about diabetes. It is beginning to seem that there's some probability that diabetes is what's called a dysbiosis, a breakdown of the normal gut microbial landscape. This ecology isn't functioning just right. Mm-hmm. And there's a belief that we might begin to be able to manipulate it. It's going to be true for mood disorders. There have been experiments that have been done with functional MRI, these are very sophisticated techniques, that can measure differences in people's way of thinking, their moods, incidences of depression, based on probiotic yogurts. I mean, this sounds like, health food crazy stuff, but it's not. This is this wonderful and weird new world that we're heading into together.
Well, one of my colleagues, Eileen Wright, who practices down in North Carolina, talked several years ago about the brain-derived neurotropic factor that was put out by the microbiome to actually get the brain to heal based on what the gut was doing, and that particular organism was contributing to brain health.
Well, two salient factoids. One is the majority, about 90% of the serotonin. This is a powerful brain chemical, serotonin. 90% of this is produced by gut bacteria. We used to think that it was all produced by our internal cells. That's not true. There's a deep connection. The gut-brain axis, neural axis, is now clearly understood. It is no longer fringe science. We're going to learn how to manipulate that in ways that are absolutely unbelievable. But we're just at the infancy, and it takes a lot of trial and error to learn how to do this properly.
Well, it would seem that the complexity of the problem would be the big issue, because then I'm starting to think, as you're talking, the different organisms affect each other. So you have not just that organism itself, but the confluence of organisms as they relate to each other.
That's a brilliant thing to say, and really a deeply informed way of thinking about it, because here's the thing that is also new and still little understood. You can make an explicit analogy between the concept of dark matter in the universe and our understanding of the microbial world. Here's exactly what I mean. Up until recently in astrobiology, they spent all their time looking through their telescopes and looking at the great, the stars, the planets that they could identify, the asteroids and the comets, and they cataloged them. But then they found out something that was really deeply disturbing. None of the equations made any sense. There was an absolutely imperative missing fraction, which they named, because they couldn't see it, dark matter. This dark matter was at first just theoretical, and now it has been proved. And it's about 90% of the total universe. Not that I understand any of it, or any of it, and maybe nobody understands it yet. But this dark matter is essential and distributed throughout the universe. Here's the thing about the microbial world vis-a-vis us as these hologenomes, these combination organisms I'm talking about. When I went into medical school, and all of my practice actually, here's what I was taught. If your culture is negative, it's sterile. Absolutely, ma'am, your urine is sterile. Your infection is gone. Babies are born sterile. The uterus is sterile. Amniotic fluid, if there's a bug in amniotic fluid and it's infected. All of this stuff was dead wrong. What we didn't understand was that culture was only identifying about 10% of all the total microbes, probably even a smaller fraction than that. 90% was our dark matter. Couldn't see it. Didn't know it was there. And so we assumed there was nothing there. Now, with a science called metagenomics, a fancy term, just means we can look at the genes that occupy an environment. And from those genes, we can know what microbes are there. We can't culture them, but we can use very sophisticated genetic assessments. This has revealed an enormous constellation of microbial life, which is also part of us and is yet uncatalogued. So getting to your comment about how difficult it is to do these studies, precisely, we have to learn how to determine what the baseline is, and we're not there yet. We have to learn how, when we've manipulated the fraction, when we've done a manipulation, how do we collect the specimens so that we can compare to the next study? All of this is new, and it'll take decades to work this out, Charles.
Well, that was my next question, because I started wondering as you were talking about it, You were saying genes, and I started thinking on a more primitive level, from a cellular level, because I was thinking cellular genes, but you're really thinking not just the cell itself, but the entire set of organisms. And
Correct. Yeah, what you have to start thinking about is and someday when we're talking about depression, of course we'll talk about the brain, and of course we're going to talk about the influence of the environment, the environment as we mean my parents didn't buy me lunch and I'm hungry, I went to bed hungry, and all those things that are absolutely imperative. I'm not diminishing them in the slightest. What we will learn to honor, in addition to those things, is what's called the full range of epiphenomena. What does that mean? All of those things that happen to us that are stresses on our catecholamines, on our neural system. What are the most consequential stresses? Microbial interactions. We're only beginning to delve into this puzzle. These interactions and the immunological rules that guide how we form reactions to those ecologies that I've talked about before, that rainforest in your gut that determines the metabolites that circulate out through your body. We're only beginning to do that. Here's a thing that your listeners might find very odd. You don't determine that you're full because you're full. This is really going to sound odd, but this is actually a fact. Your body knows it's full when about 1 billion more bacteria have reproduced in your stomach. It takes about 20 minutes, and that's why people tend to feel full after 20 minutes of eating, or 20 minutes after eating. We used to think that the circulating brain chemicals that induced us to say that what they're called satiety factors, that they're peptides, that we thought, those are from the cell lining, of our own Bill Miller cells lining my gut. No. Well, some of them are, but the great majority are my bacteria, and they are determining when I'm full. So even for things like obesity, we're going to learn how to do differently. We're also learning that the realm of infectious disease has consequences that would have been thought ridiculous years ago. For example, there's just a recent report, a pretty good report, suggesting that there's a high probability that coronary artery disease is an infectious disease. We haven't actually identified the specific agency, but we're getting to this term I use, dysbiosis, which just means a breakdown of the proper ecology, the breakdown of the cell lining ecology, which includes all players, both personal endothelial cells and microbes. They're all there, they're circulating there too. When that protective ecology breaks down, even a normal constituent of that ecology can go bad. It's a new term called pathobiont. It means in one circumstance a bug can be good, a microbe can be good. In another set of conditions, it can be your enemy. This happens in the gut all the time with C. difficile, Clostridia difficile. This is a normal gut inhabitant, but if it gets out of control, it gets out of whack, it gets to reproduce and flourish at a rate that is improper for the total ecology, then you have C. diff, inflammatory bowel disease, which is a terrible affliction and is being treated with microbial transplantation, fecal transplantation, with a very high success rate. This is the new world we're heading towards, and it's going to be exciting, and it's going to matter to your patients in psychiatry. It's going to matter at every single level, and we're just beginning to learn how to do it.
Well, now let me ask you probably a very elementary question for you, but I'm going to go ahead and ask it anyway. What is the vehicle? How do those microbiomes actually communicate? What's the system for sending signals to each other? Is there a specific system? Are there multiple systems? Do you have any answers for that one?
Well, that's a terrific question. We're just unraveling it. Here's what we do know. The microbial world in its colonial form. So when you get an infection, it's never just one microbe. Let's just say I cut myself and I get a staph in the cut. Well, I could get a couple of them, and it wouldn't, my immune system will fight them off. But if they get to form what's called a biofilm, so they get to the point where they can collaborate together, they all of their cellular intelligence can start to work together into a form that is their preferred habitat, their preferred status. If they reach that moment, now I'm in trouble. So what we know, actually, very good deep science, is been likened to chatter. In other words, microbes talk together so abundantly, it's like a vast communication system. It's going on all the time. And we're learning that communication, that level of information trading, is absolutely abundant and ever ongoing between all of our cells, all of my personal cells. They're chattering away to the microbes are chattering to the microbes are chattering with my cells. This is a complicated system. It's a gorgeous, complicated system, a new way of looking at our lives and eventually medical practice that is all new. And then, of course, it changes evolution, because evolution can't be a very simple Darwinian narrative, as pleasing as it is. It cannot be that when you start from the very beginning, where the cells themselves have purpose, have cognition. It changes it from just randomness to, certainly randomness still counts, but it changes it to a system of evolution from the cell upwards, which has direction. At each point in time, the cells are working together to try to achieve their best status. It's not just reproduction, not just reproductive success. The narrative is the cellular world trying to sustain itself according to its needs.
Bingo. Yeah, well, do you have a video on this by any chance? You are so articulate. And do you have anything on YouTube that you've done with this?
Yeah, I have. If people would like to see a brief explanation of just a few minutes, there's a link to my YouTube video on my website, themicrocosmwithin.com. And I welcome people to go there, take a chance to look at that. If they're interested in getting a lot of new information, just like what I'm talking about, but with some links to scientific articles, it's join me on Twitter at BillMillerMD.
Well, I'm going to pull that over and put it in your show notes.
Thanks.
I'll just download it and load it right there in the show notes, so they get a chance to see this handsome guy out there in Arizona talking about stuff.
Yeah, I know. Brad Pitt is he's gonna hit that video, and he's just gonna melt, poor fella.
Well, one of the things because everything you're talking about, I didn't know if you know that I'm interested in this, but the whole quorum sensing concept with the Candida was an interesting. There was a TED talk on quorum sensing, and
I didn't know. I didn't know that you're interested in that. That's great stuff.
It is totally interesting, but I know so little about it. I'm curious about it, and I'm looking forward to being educated about it. But I think it's interesting that those guys are in there deciding what they're going to do together, while you're, as a physician, trying to do something about eradicating them, and they're having these group confabs where they're deciding it ain't going to happen that way.
Well, exactly. The question, quorum sensing is very important because it shows that the capacity of intelligent organisms to act together when they're still in the unicellular form, they're in the single cell form. They haven't come together to be a multicellular organism like you and I are, but they're still single cells. And yet, what's the importance of something like quorum sensing? It teaches us some very deep principles of evolution and biology. It's all about information transfer.
That's right.
Horizontal trading of genes, which is abundant in bacteria, viruses infecting cells, bacteria, prokaryotic cells. This is communication. This is information transfer. Information transfer is what underscores evolution. And that is what the vital difference, because information transfer leads to, just as we have in human cities. Charles said, how do beings like us come to be? That's the big question in evolution, which in turn impacts healthcare. How do we get to be what we are? And the theory had always been, well, it takes a huge amount of time, and the thing that reproduces best it has a little bit of an advantage, only the strong survive. It doesn't work that way. Yeah, yes, the strong people survive, strong cells survive, strong organisms persist. I mean, obviously, you only get to reproduce if you're strong. But the greater thing is taught by quorum sensing and the unicellular form. Intelligence, cognition, cellular purposes, collaboration, communication, information transfer. It's engineering. The best way to think about evolution, the best way to think about how we are as bodies, is we're engineered to be the way we are by intelligent cellular things. This is not intelligent design. We're not talking about a creator out in the ethosphere. Everybody can believe what they want about a creator and his purposes, but what we're talking about is intelligence, starting from the very first instantiation of life. The very first moment there was life, there was cognition, there was intelligence, not like our intelligence. Bacteria are not on Skype and not doing interviews, but they're chattering together according to their scope and scale. This impacts medicine now because that's what we are. We're engineering projects at every local tissue ecology towards the purpose of sustaining the entire organism. And then what do? We recapitulate. We go back through the one cell size. Here's the thing that's not many people have thought about in evolution and in biology. Why do we have sex? Why do we go back to a single cell form? You'd say, well, scientists must have figured that one out for sure. I mean, what could be more elementary than trying to understand that? We've never had a good answer, but we do now. It suits cellular purposes. That's how they reframe their communication system. That's how they keep it tuned to the environment so that you can have survival. It's a little bit deeper than we ought to go into on this particular show, but let's just say that our current understanding that the action is all in the cellular level, and we are an enactment. We are a city. What matters in a city? It's the people that are building the roads, the buildings, the entertainment, the interchange of information. What builds a city? An exchange of information. I use an example in my book. Could any Native American in the year 500 AD, looking from the New Jersey side of the Hudson River and looking out to the future site of New York City on Manhattan Island, ever have imagined that something as complicated as Manhattan with lattes and internet would be there someday in buildings that are 175 stories tall? No, of course not. It was beyond imagination. If we use the same process of reasoning about the cellular cells and their intelligence, their collective ability to engineer environments that work for them, you could end up with organisms like us. That requires something that's beyond random, which is what cognition does. When it's invested in every cell and every microbe, you get a different endpoint, and that's our wondrous world.
That is such an interesting point right there, because when you're thinking about it, and the communication then pulls that whole group consciousness together, and there's strength in numbers, and then through that they then defend themselves better and increasingly well, because they're talking effectively with each other.
Exactly. That's precisely right.
It says something about participatory medicine, doesn't it?
Well, we're getting smarter. We really are. Medicine is adaptable. Old ideas die a slow death, but we live in a wonderful era, and I'm not negative in the slightest. We have great science. We are making continual advances, and now, if you look at the slope of the curve of our advancement, my goodness, we're on an exponential increase. It's a great time to be alive. It's a great time if you've ever needed medical care, now is a better moment than any time throughout the entire history of the planet.
And tomorrow it'll be better.
And tomorrow it will be better because we're
going to be communicating. Bill, I want to say very sincere personal thanks to you, because this has just been such an interesting conversation. Bill has provided a PDF for us Cognition, Information Fields, and the Hologenomic Entanglement. Evolution in Light and Shadow. How intriguing that is for a PDF. And, that's going to be available.
It took me a couple of minutes to come up with the title. Yeah. That's the article.
No, that was a lifetime of thought on that one, buddy. But no, that's very interesting. It's going to be available at corebrainjournal.com/033 download. And so for those of you in the car, you can pull out a very nice, and Bill, thank you so much for sharing that with us. I'm looking forward to seeing it. I'm definitely going to read the book. It's a very, very intriguing book. Thank you for taking the time with us.
Thank you for having me as a guest. It's been a privilege.
So do you have any closing, or we've said thank you, we're winding up. I just want to leave you with one last shot. Is there something that you would say that we should do as we walk away from this interview in terms of our next step at appreciating and using this information on a daily basis?
Yeah, I would say I'll add one quick thing. I'm a big believer that there's an opportunity with this new information about the microbiome and how it deeply affects our metabolism in ways that we're only beginning to understand. I believe that every person needs to consider experimenting with, try things like probiotics. I grew up in a medical culture that would regarded any kind of natural remedy as anathema. It was always considered foolish. At this point, contrary to what I was taught in school, some of these things seem to have effects individual to individual, sometimes quite profound. Probiotics, fermented foods, these are very high in natural organisms. Some people feel genuinely different with things like allergy, mood disorders. There is experimental evidence that some of this has a healthful effect. Again, it's individual by individual, so I encourage people to take control of their own health, experiment for themselves, make their own personal judgment about what works for them and doesn't.
Appreciate their own microbiome.
Exactly, and try to manipulate it sensibly. There's a good literature on the web, and they can find it.
Thank you so much again. Really appreciate it. Look forward to talking to you again sometime in the future, Bill. It's been.
It would be terrific. I enjoyed it very much. Thank you.
Thanks for listening to Core Brain Journal. We're working every day behind the scenes to bring you reports that connect research benches with those street trenches. Here we share the complexity of mind science because, as you know, details really do matter. One of the most pervasive, misunderstood challenges is how commonplace medications, like those written for ADHD, are used so regularly without clear guidelines. If you think you'd like more specifics, take a minute to download my 2-page PDF packed with video links and references on the absolute essentials of how to start ADHD medications. They're easily available at corebrainjournal.com/start. Thanks for listening. Do connect and stay tuned. Together we can make a difference.